friends, welcome back to our channel BIOZone. Myself Vidisha Bhattacharya, and today we come back with a new topic of photosynthesis. So as you all know that generally in the daytime plant photosynthesizes, right? Now how many of you knows that this photosynthesis can't be stimulated by every wavelength of the light in the visual spectra? If you don't know, please stay tuned with us, as because in today's lecture we will discuss about how a plant responds to the different wavelengths of light. So let's start. Before knowing that, you have to know the main components of the photosynthesis, that are carbon dioxide that a plant collected from the atmosphere by some special pores in their leaf, which called the stomata. The next thing is called the water that they collected from the soil through their roots. And the third one is the light, and the fourth one is the pigments. Now, where this pigment situated? Generally, if we see the cross section of the leaf, we can see inside of the leaf have lots of cells, of which one cell is called the mesophyll cell. Within this mesophyll cell, we have a bilayered organelle called the chloroplast. Now, these chloroplasts have two portions. One is the liquefied region, this is called the stomata, and coin-like structure which is piled together is called the granum. Now, each coin of this structure is called the thylakoid, within which the pigment resides. Now here we have two types of pigments. One is called the main pigment, that is in the case of plant is chlorophyll A, and the accessory pigment that helps the main pigment to excite is generally in the case of plant is chlorophyll B, xanthophyll and carotenes. Okay. So generally when a light falls in a certain region, what happens? It spreads, right? Now the accessory pigments collects the light's packet and transfer it to the main pigment so that the main pigment can excite and can respond for the photosynthesis. Now here one question may strike on your head, that why the two forms of the chlorophyll are acting differently, right? For that, you have to understand the structure of the chlorophyll and how they are responding to the different wavelengths of light. So first, we will discuss about how these pigments works and respond to the different wavelength of light. So generally, when we come to know that light is important for the photosynthesis, one question strikes on the head of scientist Engelmann. He thought every visible spectra can work equally for photosynthesis or not. For that, he conducted an experiment. Now, in this experiment, what he does? He uses three components. One is a white light, that is a polychromatic light, as because the white light is a mixture of 7 wavelengths of light that produces 7 different color, what we can see in the rainbows, right, which is called a VIBGYOR. Now V stands for violet, I stands for indigo, B stands for blue, G stands for green, Y stands for yellow, O stands for orange, R stands for red. So this 7 color mixture is the white light. The white light can splits into these 7 different colors. Okay. So the next component is the prism that can split the white light into the monochromatic light. That means in different wavelengths of light having this respective 7 colors. And a jar he uses that contains green algae, Spirogyra, which is submerged in the aerobic bacterial solution. Okay. Now what he does? Generally, he uses the white light that was passed through the prism. Now when the white light strikes on the prism, it will split the white light into 7 different spectral range. Now these 7 different light strikes on the different region of the jar and illuminate the jar. Now what he saw from here? He saw that the aerobic bacteria are accumulating in the blue light and the red light range. Now why the aerobic bacteria are accumulating? Generally we all know that the end product of the photosynthesis is oxygen and the glucose. So in the case of green algae, same is happening. They utilizing the light and producing the oxygen and the glucose. Now aerobic bacteria as uses the oxygen for their activities. So where the more oxygen is producing, they are accumulating in that region. So from here first concept comes that all wavelengths of light is not equally responsible for generation of sufficient amount of photosynthesis. So from here a concept comes which is called the action spectra. That is the rate of the photosynthesis which is correlated with the amount of light that a plant gathers for performing the photosynthesis process. Now when we are talking about the spectra, we generally uses 2 terms. One is called the action spectra that we discussed and the absorption spectra. Now what is that? Generally the amount of light that a plant accepts or the average amount of light that a plant can consume is called the absorption spectra. And the action spectra defines as the rate of the photosynthesis which is influenced or stimulated by a amount of the spectral range or a number of the spectral range. Okay. Now previously it was thought that this action spectra and the absorption spectra is similar, but several scientists proved the process by the spectrophotometric analysis that these two are quite different, which we can see in the graph also, right? Generally, the action spectra defines the rate of producing the oxygen by the process of photosynthesis, and the absorption defines the average amount of the light that a plant consumes. They showing their peak in the blue and the red light range, right? But their graph is not same. This showing an overlapping graph, right? In the case of the blue light, what we can see that the absorption spectra is much higher in respective to the action spectra. And in the case of the red light, we can see that the action spectra is much higher than the absorption spectra. So this is a basic difference between the action and the absorption spectra, what we can see in the graph also. So depending on this and depending on their acceptance of light, so in the different region, we differentiate the accessory pigments and the main pigments. Okay. So what we can see in the next graph is the chlorophyll A is sufficiently producing their peak in the blue zone and the red zone, but in comparison to the chlorophyll B, it producing more high peak in the red zone but not in the blue zone. So next is the carotenoid that showing the peak in the central region, what we can see here near the blue zone. So from here we can see that the main chlorophyll is responding more in the red light than the chlorophyll B, that is the accessory pigment. As we can see in the previous graph also that the action spectra is much higher than the absorption spectra in the case of the red light. So from here we can conclude that the chlorophyll A can be the main chlorophyll. Apart from that, if we look at the structure of the chlorophyll A and the B in the picture, we can see that the chlorophyll A and B look similar, but in the second ring, we can see that they are differentiating from each other. Where it is differentiating? Generally, in the second ring, we can see that the chlorophyll A contains CH3 group and the chlorophyll B contains CHO group. For these single differences, they works differently, and chlorophyll A becomes the main chlorophyll and chlorophyll B becomes the accessory one. So if you understand our video and if you like our video, please subscribe to our channel and stay tuned. Ta-da!